Hello, everybody. I'm Mike. I'm happy to introduce Dan Brown today. Dan worked on the human and mouse genome projects at Whitehead, and he's now faculty in the bioinformatics group at the University of Waterloo. He's going to be talking to us today about haplotype inference, and I'm just going to pass it over to him now.
Thanks, Mike. It's nice to be here. I think I'll start with this here for now, but I may start walking back and forth, which will probably drive this fellow nuts. So I'll try to be good about standing still. Let's see. All right, so I'm Dan Brown. It's nice to be here. I'm visiting you here right now from UC Davis, which is where I'm on sabbatical. Normally I spend most of my time at the University of Waterloo, which is in southwestern Ontario. I put up the full affiliation there because, in fact, our department was recently named after David Cheriton, who was actually an early investor here at Google. And so he used some of his money that he earned from that to endow his alma mater, which is kind of exciting for us. I want to start off with the obvious quote, which some of you have probably seen, which is a quote from the Kitzmiller versus Dover trial. This was the intelligent design trial last fall in Dover, Pennsylvania, where the question was whether or not creationism should be taught in public schools in that community. And the quote here is from Robert Pennock. He's a philosopher of science at Michigan State, and he says that you can get a job at Google if you know something about evolution. They're looking for people who know something about this. I should point out that I don't need a job, but nonetheless, it's nice to actually come to Google and have the chance to spend a little bit of time telling you about evolution. So I want to start off with a problem. You may not see this as a problem, but I don't look like you. You may see this as a terrible problem, and if so, that's very flattering. But it's an interesting fact about human beings that we don't all look the same, that we don't all respond to stimuli in the same way, that we in general, despite being very genetically similar to each other, are not in fact 4 or 6 billion clones of the same genetic material. Rather, instead, because of a variety of different things, human beings respond differently to different stimuli. This is true in the fact that we don't all look the same. It's also true in the fact that we don't all get the same diseases for the same reasons. Now, there are lots of reasons for that, of course, and the majority of them are not, in fact, genetic, probably, or at least many of them are not, but some of them do have genetic bases. I don't look like you in part because of my genetic upbringing. Now, there are other aspects of the reasons why I don't look like you, ranging from the decision I made today to wear a black t-shirt, all the way on up to the way I was fed when I was a child, and so on. But nonetheless, some of why I don't look like you has to do with my genetic makeup and your genetic makeup. And in general, while why I don't look like you may not be of great influence, or may not be of great interest to you, it's still the case that we should know why it is that different human beings respond differently to different stimuli, at least at a genetic level. And the way that we do this in biology is through association mapping, which connects particular traits with particular genetic variations. Now, this is pretty easy at this point for a variety of different things. It's easy for things like breast cancer susceptibility, where the BRCA genes have such an impact on that susceptibility. It's especially easy for monogenetic diseases like cystic fibrosis, where all you have to do is identify a specific gene which is responsible for the vast majority of the origin of the disease. In fact, at this point, if you created ex nihilo some new genetic disease that no one had ever seen before and suddenly found a community of people who had it, we could probably identify the gene responsible for that one gene genetic disease in a matter of a few months for a couple $100,000. This is a remarkable change over even 10 years ago. But in fact, most diseases that actually affect people, most of the explanations for why it is that people are, say, obese or diabetic at a genetic basis, aside from their lifestyle differences or whatever, are multifactorial. It's not the case that single genes explain those diseases. Rather, it's the case that nowadays we have to look at a connection of many genes acting in concert as being the genetic makeup that's responsible for a particular disease. So there's basically two problems that are involved in the difficulty with doing association mapping, with connecting diseases and traits to genetic bases. Now, the first one is actually the most important problem in all of science, or the most difficult problem in all of science. And that's the problem of relating correlation with causation, of course. If you say, I looked at a collection of 500 people and they all had this common genetic variant and they all had this disease, and then I looked at 500 other people who didn't have that common genetic variation and didn't have that common disease, that's interesting. In fact, it's fascinating potentially, but it doesn't tell you the first thing about why it is that the 500 people with the genetic variation got the disease, or why the ones that didn't have it didn't get it. So of course, that's the most basic problem of all of science, though. Why is it that some connection, some correlation, how does that conflate with causation of a particular trait? This talk is not about that question because, of course, this talk is not the end of science. Instead, this talk is about the other problem that's also difficult, which is just looking at a collection of individuals and trying to identify what the genetic variations that they have actually are. In other words, if you look at a population of different people, asking the question, for particular genes, which variations on that gene did different people inherit from each of their parents? That problem is called haplotype inference. It actually goes by lots of different names, but that's the name I will give it in this talk. And it's the problem of separating out parental chromosomal inheritance from each other in one person or in a collection of people. So this really does matter. Even inside one person, it actually matters. And I'm going to spend a little bit of time now explaining why that's so, why it's even important to identify for a particular person what that person inherited from their father versus from their mother. And I'm going to do that by telling you actually the origin of this field, which, embarrassingly enough, I only learned about a few weeks ago when I was talking with a statistical geneticist at UC Davis. So each person in this audience has 2 copies of human chromosome 6. You inherited one of them from your mother and you inherited one of them from your father. Now, on that chromosome are a collection of immune system genes called the major histoincompatibility complex. You probably inherited something different from your mother than what you inherited from your father. The reason why is just that there's a lot of different variations on that chunk of DNA that are found in the entire human population. And unless your parents were, say, brother and sister, which would be a very bad idea, you probably inherited something different from each of them. Indeed, actually, it's reasons having to do with, for example, that are among the many reasons why, well, for example, having children with your cousins is perhaps not such a wise idea. So you got something different from each of your 2 parents. Now, suppose that you got very sick and needed a transplant. Suppose you needed a liver transplant, for example, and some liver got put into your body, and that liver was from a person who had a different kind of genetic basis that they had inherited from their parents. Well, then you're in something of a problem, because if you were transplanted with an organ that didn't match either of your parents exactly, rejection can happen. So the trouble with that is that the technology that you can get to identify somebody's DNA sequence doesn't actually separate the maternal chromosome from the paternal chromosome. It doesn't identify what came from your mother and what came from your father. So let's look at this simple example here. Here I've got 2 different DNA sequences that might have been inherited in one person. Remember that DNA is a DNA sequence is a very, very long sequence of letters over the 4-letter alphabet A, C, G, and T. At least as far as we're concerned, that's what it is. Obviously, it's really a molecule, but thinking about it in the sequence abstraction is perfectly fine for us. So suppose that you've got something like that, and suppose that a person is needing a transplant, and that those are the 2 things that they inherited from each of their parents. From their mother, they inherited in a particular chunk of sequence AGTAT, and from their father, they inherited GGTTT. So if you look at the 2 columns that are indicated in red there, you'll notice that those are the 2 columns where the 2 parents differ from each other. The mother in the first case has AA, and the father has GT. Now, the trouble is that the technology that does sequencing doesn't distinguish which came from what. And so, in fact, if you just used a typical DNA sequencer on somebody whose sequences were instead GGTAT and AGTTT, you would actually get the exact same outcome. Because in the first column, you've got 1A and 1G. In the 4th column, you've got 1A and you've got 1T. And the other 3 columns are the same in both. So in both cases, you wind up with the exact same result off the sequencer. But unfortunately, suppose that you were actually the person on the top there, and you were transplanted with an organ from the person on the bottom. So you actually have the AGTAT haplotype, which is what we will call that chromosome, and you get transplanted with somebody with the GGTAT. Well, you could die. I mean, you actually really could die. This is going to cause potentially rejection, and you could die. So in fact, the question of separating out the two chromosomes, identifying for a person exactly what they inherited from each of their two parents, maybe not worrying too much about which one was mom and which one was dad. That may not actually matter very much. But identifying the full sequence of what they inherited from each parent is actually vital because, again, if you get it wrong, you could die. There are not that many places in science, and certainly not that many places in computer science, where we wind up saying to ourselves, wow, this could actually result in somebody dying. But this actually is one of them. Now, it's also the case, separating away from this question of somebody dying, there are also just reasons why you might wonder in general. So it could be the case that a particular effect, a particular phenotype, a particular experience in the real world might depend on having the A and the A together, or the G and the T together, not, say, the first G with the subsequent A or the A with the T. And so being able to separate out those two chromosomes might be useful just for the question of joining together effects from two different variations into one explanation. So the question of haplotype inference there then is the question of taking apart, of identifying from what came from the DNA sequencer and separating out the two chromosomes that explain that. So again, that's what these problems are all about. And there are a whole bunch of different problems that all have sort of the same flavor, but all differ a little bit in structure. And they're all of this form. You're given a collection of sequences, a collection of DNA sequences that come from two chromosomes, where you have some ambiguity at certain sites about which one came from which parent. And the question is to find the two parental sequences, the one that corresponds to the mother and the one that corresponds to the father, that explain either that one person or that collection of people. Oftentimes, this is actually just cast in simpler terms because biologists know this language well enough that they don't have to worry about it in terms of sequences and chromosomes. They just will say that the input are genotypes. Those are those sequences that have the ambiguity. Is this an A that goes with an A or an A that goes with a T? And how do you distinguish which from which? The input are those genotypes, and again, the output are the haplotypes, which are the chromosomal sequences separated from each other. And that's the domain of this talk. There's a lot of different problems of this type. Again, they actually go back quite a long time. They go back quite a long time because when transplantation was first started, people needed to be able to identify, and they did it using antibodies actually, for a particular person, which two types they belonged to. This is much more complicated and much more important since, again, you could die, and transplanting is really expensive, so it's kind of stupid to waste all that time and then have the person die for rather dumb reasons, than, say, blood typing. But still, nonetheless, that was how it was started. Nowadays, we'd instead be just doing it off of sequence. And suppose that, here's a simple example of one such problem. Suppose that you're given sequences of two different population members, and in this picture here, the positions in the sequence where there are no variations, where there's only one element in a particular position, both parents agree with each other. The only places where there's any ambiguity are the ones where I've put two different symbols over top of each other. So suppose that you've gotten a couple members of a population, and you've taken genotypes of those people, and the question is to try to figure out what goes with what. What was the right way to explain it? Now, in the case of this simple example I've given you here, one thing to notice is that you can actually explain both of these two population members using the same haplotype, one in common. And it's that ATG-GTT haplotype I've drawn there. Assuming that I've done this right, it's in red in both cases there. That could be the sequence of a parent of one of those people and also of a parent of another one of those people. Now, why does that make any sense? Well, here's why it makes sense. It's certainly a bad idea for you to have children with your sibling, but it's certainly reasonable to assume that your sibling might have children and so might you. And so if you're looking at a family, it's reasonable to assume that, in fact, multiple members of the next generation of a family will have inherited the same haplotype from their parents. So if, for example, this is, these are the sequences from two brothers, that actually kind of makes sense. They might have had one parent in common, and they might have both inherited the same haplotype ATG, GTT from that same parent. So this question of trying to separate out the two confounded sequences for every population member, in fact, asking how to do that, there's no way of doing that when you've only got one member of a population. But if you're looking at a collection of people, well, you might be able to do it just by looking at this kind of reuse. So just to now give you, that's sort of an introduction to what we're going to talk about today. Now I want to give you a little bit of I don't know, some abstraction that makes this feel a little bit more like the computer science problems that at least I, as a mathematical computer scientist, feel a lot more comfortable with. So again, we start off with genotypes. These are these sequences that come off of a DNA sequencer, but they have this difficulty that if a person is sequenced and a particular position of that person's sequence differs in their two parents, well, we don't know what came from which parent. Haplotypes are actually a lot more important than genotypes if we're asking about inheritance of complex diseases or trying to avoid transplantation rejection or such. And again, now let's just start thinking about this problem rather than as some ambiguous kind of fluffy biology problem. Let's get down to brass tacks and think about this like a math problem. So I'm going to start simplifying the problem I just told you about now. I should also say, I'm happy to, if I'm snowing you, please stop me and just ask a pile of questions. That will make me happy. Let's step back away from thinking about DNA sequences, like in this picture, and let's just go to math instead. And let's, in fact, in particular, start abstracting away the DNA sequence entirely. I'm going to assume that I'm now going to restrict the problem domain to places where, if I'm looking at a collection of DNA sequences, at least one of them has ambiguity. So for example, in this here, every one of the haplotypes that's apparent for either of those two people has a T in the second position, because the second position in both of those is TT. So I don't even care about that particular position. So just forget about positions where everybody has the same thing and focus on ones where there's at least a little bit of ambiguity. The second assumption that I'm going to make is that at every variable site, I'm only going to let there be two different population choices. So for example, here, I'm not going to allow the first position of another population member to have a C as a choice, because I've already got A's and G's. And I'm just going to make the rule that every position where there's variation, there's only two common types. That actually is true in the real world. What's more is actually, if it turns out that you wound up finding a rare case where that wasn't, we can actually deal with that anyway. So we're going to make the assumption that there are only two choices at every site. And for simplicity, rather than A, C, G, and T, which is a bit of a pain, let's be mathematicians and use the binary alphabet instead and have just be zeros and ones. And then we just need some notation. And the notation that we're going to say is, now I'm going to have a matrix as my input. That matrix corresponds to telling me what is in a particular position of a particular person's genome. If a person has a 0 in that matrix, that corresponds to saying that both of their parents have what we're going to call allele, or I'll just call it value for this talk, value 0 at that site. If both of the parents at a particular site in a particular person's genome have value 1, I'll stick a 2 in that matrix. Those are all the ones that are really easy to resolve, though. I mean, everywhere you've got a 0, both parents are the same. I mean, that's the cases like the first column of the first person there. You know what both of those are going to resolve to. The ones that are more interesting are the ones where there is this ambiguity, and in those positions we're going to put the number 1. Now, I should say, if there actually is anyone in this audience who's ever worked on this problem before, I'm totally abusing notation. I'm taking advantage of the fact that probably most of you are innocent of this problem and cheating. The standard notation for this problem that biologists use reverses the meaning of 1 and 2, and I'll come back to why we've made the change of doing it this way in just a second. Yes?
So when you go back to the previous picture,
This.
yes.
Mm-hmm.
So you're trying to find two haplotypes, the first person, two haplotypes.
Yes, that's right. But the thing that actually gets us some win here is the fact that we might share them. In fact, if you had random garbage from all over the world and, bacteria well, bacteria don't have this problem because they don't have two chromosomes because they don't have two parents, but some organism that had two parents that you'd sampled from all over the world and there was very little genetic relationship between them, you'd have just as many problems as before. The thing is that at least if you have some family structure, maybe you can share haplotypes or have haplotypes that look similar to each other because, two people might have related parents. There might be one or two mutations that differentiate them, but they'd at least be similar looking to each other. But that's right, we want to find two parents for every population member. Good. Okay, so that's the problem. We're given this matrix. Just to think about it, I didn't draw it here as a matrix because, well, it just didn't really fit on the slide, but the problem that we had before, we can imagine casting in this term just like that, where the first guy, we're going to have all of his places where there's only one choice be 0, and that means that the A in the first column turns into a 0, the T turns into a 0, and so on, where the only thing that's different, and places where there's choice are 1s, the only thing different in the second guy is you'll notice that 2 in position 3, that's because he has 2 Gs, and maybe that's the allele number 2, or that's the second allele number 1. Having 2 of those means that you have a 2 in that particular entry. And the places where there are 1s, just to give you a little bit of terminology, those are called heterozygotic positions. That just means that at a particular site in the genome, you inherited something different from each parent. So again, all that we have to worry about here is figuring out how to arrange the 1s. In every 1 position, there has to be 1, 0, and 1, 1. The only question is which chromosome has which 0s and 1s so that you put them together and they all line up properly. In the positions where you have a 0 or a 2, it's all very easy. You've got to have a 0 in both parents or a 1 in both parents. The reason I should say now that we use this notation, which cheats against the standard notation here, but at least to me as a mathematician just feels vastly simpler, is that now in this abstraction, a genotype is a sum of 2 haplotypes. You're going to be taking 2 vectors, adding them, 2 binary vectors, adding them together, and producing this vector over 0, 1s, and 2s. And, well, that just makes me happier. So now life is much simpler. And in particular, if we have a population of n population members that we sampled from, and we sampled them at m, as in Mary, different sites, we have an n by m matrix over 0, 1, 2 is the input. And the only question is now, how do we decouple those m rows into 2m, those n rows, sorry. How do we decouple those n rows of genotypes into 2n haplotypes? So that if you add 2 consecutive rows up in the new matrix that we're going to form, you get 1 row from the original matrix G. So again, the input is an n by n genotype matrix G, n population members, m genome sites. And the goal is to find a 2n by m haplotype matrix H, so that if I add up 2 rows of H, I get 1 row of G. Now, the only question here is, well, I mean, think about it. If you've got an input matrix G, the number of explanations for any row that has E 1s in it is 2 to the E. Because it's just a matter of, do I put a 0 here, or do I put a 1 here? Do I put a 0 here, or do I put a 1 here? You can divide that by 2 if you say that you can't identify parents from each other. So if you can't separate mothers from fathers, then there's one that explains at least half of it. But you still have an exponential number of such explanations. Which one is right? Well, that's an interesting problem, and it certainly lets people have arguments as much as they want to. But one way of thinking about this is to actually do some evolution experiments. And to say, the way that I'm going to answer that problem is I'm going to say, I have a model of how these sequences could have evolved. The sequence is in H, my haplotype matrix. I have a model of how they could have paired together, which in evolution we say that people mate rather than pairing. A random mating model for explaining how the haplotypes paired to form genotypes. And then the question is to find the explanation of the input matrix G, our genotype matrix G, that has highest probability. Now, any of you who've ever done stochastic optimization know that that's really not fun. I mean, you've got this very complicated model of evolution. You need some way of really explaining how the mutations happened, how recombinations happened, how starting from some least common ancestor, all of these sequences came into being. And that's really nasty. And then on top of that, you have to have some way of associating those population members in H to form G, so that then you can undo the whole process and wind up explaining your genotype matrix G by H. This is awful. You can sort of split the difference if you really want to. You can try to just find the haplotypes and not worry about how it is that they paired to form the genotypes, or not have a model of how they paired to form the genotypes. That's fine. You could actually give up on the model of evolution and just focus on the pairing. That's actually easier. I'm working on that kind of problem right now. But all of this is really painful and nonetheless important. Again, remember, go back to the beginning of this talk. You could die. This is a very important topic to get right, because you really don't want to kill people. So what do you have now? You've got a population of people. You want to explain their genetic origins, and you've got a whole bunch of statistical problems that are based on complicated models of evolution, which I should point out are really hard to parameterize, really hard to optimize, really hard to do everything with in the first place, and then you have this nasty pairing on top of it. So, well, what do you have then? Well, you've got a problem, and so being theoretical computer scientists, people came in and said, why don't we solve this the way that we solve other problems that are hard, by throwing out all the hard part and just focusing on something that's similar but fun. And in particular, I'm going to tell you now about the simplest possible version of this problem, and a little bit that you can actually do with that problem to genuinely solve it, rather than tearing your hair out thinking about the statistical problems. So again, we have this input matrix G. That's our genotype matrix. It's this ternary matrix where zeros and twos are easy to resolve, and the only question was what to do with the ones. And then our output matrix is some haplotype matrix H that optimizes some combinatorial objective. Or maybe it optimizes some combinatorial objective where we restrict the class of matrices H that we're allowing ourselves to use based on something that we know about the population structure. And I'll come to that in a second. The simplest possible example of such a problem is the haplotype inference by pure parsimony problem. Haplotype inference by pure parsimony? Well, this is a problem that was introduced by Dan Gusfield, and the problem here is just have the answer have the fewest haplotypes possible. Why? Well, go back to this. Again, we wanted to reuse some of the matrices, or some of the vectors that we were using. We wanted to say that certain population members might have lots of offspring, or might have lots of related members who are having children. And so consequently, we might just want to explain our genotypes, explain our matrix G, the simplest way possible, with the fewest haplotypes. Again, this problem was introduced by Dan Gusfield in a conference paper in 2003, and that's actually where I first heard about the problem as well. So the HIPP problem, the haplotype inference by pure parsimony being, oh, the heck with all of these evolution models, I can't even think about them, they're just too complicated. Just have as simple an explanation as possible. Fewest haplotypes. And so here's a way of stating that as just a simple algebra problem. Given as a ternary matrix, n by m, goal, find a way of producing a 2n by m binary matrix where, again, pairs of consecutive rows add up to rows of G, where the number of unique rows of H is minimized, or the number of distinct rows of H is minimized. So now it's just an algebra problem, right? I mean, all you need to do is some way of structuring this addition and should be easy. Well, all right, the problem is NP hard. That's a paper, or that's a result that Gusfield cites in his original paper as being due to somebody else. It's a fairly easy reduction to prove. So just the ability to take this problem and abstract it into algebra didn't actually necessarily get you very far, but again, we're at least, we now have a real problem that doesn't make you want to tear your hair out because of all of this awful statistical, stochastic ick. Again, you can also limit the problem a little bit as well. Here's one simple limitation, and I'm not going to go into very much detail, but as I said before, you might want to make restrictions on what kind of matrix H you would allow. So for example, you can restrict H so that rather than having rather than being any binary matrix, H might be restricted so that it has to come from an explanation you could produce off of an evolutionary tree. We'll come back to what that means in a little bit, but just for now, imagine that the modern sequences that form H, the haplotypes that we're looking for, are the leaves of an evolutionary tree, and we track back those leaves back to a single common ancestor and put mutations on that tree to explain the population, or to explain the various columns of the matrix H. In any event, we'll come back to that in a second. So if H can be derived from a tree in that way, it's called a perfect phylogeny matrix. That's actually a definition that goes back also 20 years. And the question of trying to find the smallest explanation in terms of having the fewest unique rows, where you also make this restriction that you have to satisfy a perfect phylogeny matrix, that's called the min PPH problem, because it's a perfect phylogeny haplotype matrix and you want to minimize something. This problem's also NP hard. That's a paper by Bafna and Bansal from a couple of years ago. And ironically enough, it's NP hard. Lots of these problems are even NP hard to approximate. But as we'll see, they're actually not so hard to solve, which is perhaps a little bit of a surprise. So why would we use a combinatorial objective for this problem again? Well, again, the reason why we're doing this is because all that statistics was just awful. I mean, it's just painful thinking about models of evolution and how to pair things together, and it's just painful. Instead, we just have this simple combinatorial objective. Count the number of unique haplotypes, make it as small as possible. Good. That's one reason. The other reason to use this combinatorial approach is, do you really believe these models of evolution? Well, I mean, none of us is an evolutionary biologist, I don't think, but having been a little bit closer to these problems maybe than most of you, I don't. They're nice things to think about. They're good representations of evolution in the absence of selection, in the absence of of genetic drift, in the absence of a lot of the other classical things that make evolution actually work. And so why trust them to the extent of being the answer to your question? So maybe actually reducing the number of haplotypes and having that be your goal might be a reasonable approximation to a decent evolutionary objective instead. And plus, again, nothing succeeds like success. You can at least solve it. So that's the sort of practical reason to think about these problems. The other reason to think about these problems, and frankly the reason why I think about these problems, is they're tasty. I mean, think about this problem I just presented to you. I just presented to you a really trivial algebra problem at its base. And it took me 20 minutes to get you to an algebra problem, but nonetheless, all that's what it is. It's just a ternary matrix that you're saying every row of that ternary matrix is a sum of two binary vectors. Find the smallest number of binary vectors. What could be cooler than that? And there's a lot of other structure like that that's very classical graph theoretic structure that you might be wanting to look for. So all of these things are just, it's an undercolonized problem domain. There's not, despite the fact that there are dozens of papers on this area, it's still a fairly young field, being all of 3 years old. And it's just a delicious place to do work. So that's another advertisement for this, rather than necessarily why a real biologist would care about these problems. So what's not to like? And again, it does have a useful application. So that means that at night you can go home and feel good about what you did that day too, which is good. So where are we now? Well, now here's where we are. We talked about why genetic diversity is nice to know something about. It's nice to be able to characterize what separates the individual inheritances of a particular person or collection of people, because it allows us to identify susceptibility to inherited disease, answer questions about simple things like transplant rejection, and so on, very straightforwardly. In order to do that, we need to be able to separate paternal and maternal DNA in population members, which will represent by genotypes. And then again, this haplotype inference problem is the problem of separating out a genotype into two haplotypes that mutually explain it. And now we've been seeing that for reasons that are not especially convincing, but still worth justifying, you might want to study this combinatorially instead. So where are we going and what's left of this talk? Well, I've given you an objective. I've given you a nice algebra problem, but I haven't told you how to solve it. So we'll talk about how to solve it. I'll tell you a little bit that actually about why You might care about this goal because solving it is actually good for you. And then I'm going to just tell you the surprising fact that the algorithms I'm going to tell you about, which are exponential time algorithms for this problem, actually solve just like that, and maybe try to at least begin to tell you why that's true. Unfortunately, to do this, we're going to need to talk about how evolution works regardless of whether we wanted to or not, and I'm just sort of putting that off to the end. So again, we've got this haplotype inference by pure parsimony problem. It's NP hard. We're trying to find the smallest set of haplotypes that explains my matrix G. Dan Gusfield in 2003 said that the simplest way to solve this problem that he could come up with was just to do it using integer linear programming, which is, of course, a classical optimization technology for ACK. I don't know how to solve this problem. It's NP hard. It's NP hard. Maybe if I represent it in some way that has to do with satisfiability or linear constraints on integer variables, maybe that'll at least help me understand it better, and heck, maybe it'll even solve. Gusfield said that the way to represent this problem is to, and again, just a reminder again, integer programming is representing the goal of the problem, the thing that you're trying to minimize or optimize, as a linear function of a collection of variables, and then casting the requirements of your domain as a collection of linear constraints on those variables, where the restriction is that those variables have to be binary or integer. So the way that Gusfield addressed the HIPP problem was to notice, as I said before, that if you've got E1s in a particular genotype, so you've got a genotype with, say, in this case, 2 different 1s in it, that there are 2 to the E minus 1 possible distinct explanations for that haplotype. So let me detach that. It's really rare that I actually have to use microphones. I'm actually quite loud, but I'll try not to shout into this thing. Here we see that there's 2 different explanations there for that genotype, and the question is just which one what does the first 1 pair with? Here it pairs with another 1, and in the one on the right it pairs with a 0. Otherwise, these 2 haplotypes, these 2 explanations are identical to each other. Notice that there's that 1 in the 4th position in both population members, or in both haplotypes to explain the 2. Yikes. So if we start off with an explanation, or if we start off with a genotype with E1s in it, again, there are 2 to the E minus 1 different pairings that explain that. No big deal there. And again, also, we have to pick one of those explanations, and if we pick it, we have to include in our collection of H both of the things that we chose to pick it, or to explain it with. That's just fair. If you're going to explain something this way, you gotta pay for it. So that's the restrictions that are built into Gusfield's integer program. So what we're going to have then is we're going to have, for every possible choice, every possible haplotype H, that could explain any of the genotypes in G. So any binary vector that could be an explanation for any of my genotypes in G, we're going to have a variable xh, which is 1 if we choose to include it and 0 otherwise. And we're just going to count the number of those x's that we set to 1. Now think about the explanations for our genotypes. We had 2 to the E minus 1 different pairings that explained every genotype. If I pick a pairing, again, I have to pay for it, which means that if I choose to explain a genotype with explanation eta, which is haplotype 1 and haplotype 2, I better pay for haplotype 1 and haplotype 2, and I'll do that by having a variable y eta that tells me if I chose that explanation, and then the restriction that if I chose it, I have to pay for it. And in fact, this is all Gusfield's integer program is. The part in the red is the important part, otherwise that's of course that's just words. We want to minimize the total number of haplotypes chosen, subject to the restriction that we explain every genotype. That's that summation constraint that's second there, which is that we have to have chosen at least 1 explanation for every genotype. The second one or third one won't matter. And then if we've chosen a particular explanation, we have to pay for it, which is the restriction that's embodied in that 3rd and 4th line there of saying that the y variable has to be less than the corresponding x variables. Now, think back to what we talked about before. There's this exponential number of explanations. So in fact, this is an exponentially big integer program as well. This is worrying. I mean, why would we want? We've got this giant integer program, and integer programming is notoriously complicated and slow and confusing. And well, why not at least give it a shot? I mean, the problem is NP hard to begin with, so we shouldn't expect it to solve very quickly anyways. So we've got this exponential size integer program, huge number of potential variables, huge number of potential constraints. Well, try it. And it really works well. What does really well mean? Well, Gusfield did experiments on reasonably small instances of the problem, admittedly, the ones that he could actually write down, because it's an exponential sized program, and if you can actually enumerate out the program and do a couple of tricks to make it faster, then he just handed it off to a commodity program, admittedly an extremely impressive one called Cplex that solves integer programs, and they just solved. Well, all right, what does really well mean? What does just solved mean? And it also solved in practice. What does that mean? Well, despite ourselves, if I'm going to say that a particular algorithm for an NP-hard problem usually does really well, I need to tell you something about what domain I'm working with then. I mean, I can't just say, this works really well. I have to tell you, well, what kinds of problems did you run it on? And in order to do that, we kind of need something about what G is, how we got G or how we got H and paired H to get G or something like that. If you were feeling really naive about this, you could just say, oh, pick G uniformly at random over all ternary matrices. But that's not going to work, because then you'll have no sharing, and it's just a bad idea. That makes no sense from the biology of this problem. It's fine as an algebra problem, but it makes no sense in the domain. Much more sensible is to start off with some way of picking H from some distribution and then randomly pairing together the rows of H to get G. And just pick two members of H uniformly at random, pair them together and get G, and then that have, in some sense, cast your difficulty onto the question of how you make H. Again, you don't want, if you do that, where you're pairing up random vectors, you don't want those vectors to necessarily be uniformly distributed at random either. And the reason is because people have family trees, and so do chromosomes. So the way to do this is to actually have some model of evolution that says sequences evolve from a common ancestor. In this case, I'm going to say that the common ancestor has sequence 0, 0, 0, 0, 0. And the way that this works is that the children, or the leaves of this tree at the bottom, correspond to the rows of H. And the places where I've drawn X's on the tree, those correspond to single mutations at various sites on the genome. And all that you see there is that places that are descendant from an X, places descendant from a mutation at a particular numbered position, have a 1 in that position, and those that aren't don't. So for example, everything that's now highlighted there descends from that X on number 1 and has a 1 in position 1 of its sequence, whereas the guy on the right doesn't. And that model of evolution, that in fact is the that is the perfect phylogeny model of evolution, that you start with some common ancestral sequence, and then descendants of mutations have a unique, include a 1 in that position, and those that don't. So, all right, again, I told you that if you run Gusfield's integer program, it just runs, it just solves. This is not supposed to happen. You're not supposed to just push return and have it run in half a second. We now know something about at least a model for evolution and how to pair things together to make G. The way that we actually specifically produce H is we just basically run an evolution simulator on a particular model of evolution called a coalescent simulation. It picks the topology of the tree, that is to say, how the leaves coalesce as you go up towards the root of the tree, from a standard distribution, throws mutations at that tree, each independent of the other, and then that gives you your structure of H. Pick H that way, pair together random rows of H to get G, run Gusfield's integer program, bam, it works. Less than a second. So why? And in what's left of this talk, I want to just give you a tiny flavor for why. Because in fact, this is kind of a strange thing. It's rare that we get an NP-hard optimization problem that always solves, and it's even rarer that we get an NP-hard optimization problem that always solves for which we can actually explain the answer to that question, and that's what I want to finish off by telling you. So again, we've got an NP-hard optimization problem. When we pick input for it from a particular sensible distribution, run an exponential time algorithm on it, we wind up actually generating an answer very quickly that, in fact, it even turns out to be the right one. What's the structure? What's the explanation for the problem? Well, I've been trying to tell you about algebra this whole talk. I've been trying to have you think about this problem as just being about matrices. So let's go one step in toward that. And in particular, I want you to think about this problem as now being a little bit different. G is just like before, but now H is not a 2n by m matrix. It's a k by m matrix, where I've got k distinct haplotypes, and I'm pairing two members of that matrix together. A way of explaining that pairing is through a pairing matrix P, and saying that the matrix G that we're interested in is just P times H. Oh God, linear algebra. Well, too bad. I'm afraid that, you know, into every talk, some linear algebra sometimes must fall. Again, we've got two different rows of H that we add up to make one row of G. That means that P is a matrix that has one row of P for every row of G. It's all zeros except for two ones. Or actually, it can be a 2 in one position also. That means you've got two copies of the same parent. And that may seem scary until you start reminding yourself that plants can self-fertilize. So we want the smallest binary H that explains our matrix G with a proper pairing matrix P. So G is P times H. It's our pairing matrix times our haplotype matrix. And here's a fact from linear algebra. H has to have at least the rank of G distinct rows. Why? Well, G equals P times H. P tells us how to add up rows of H to make G. If G has k linearly independent rows, H has to have k linearly independent rows too. So the rank of G, which you can compute in polynomial time, you learned that in college, or even high school probably, you can use that as a way of lower bounding the number of haplotypes you're going to need. It's just an easy polynomial time lower bound. So you're going to need at least rank of G distinct rows to make this work. So is that going to be the optimal number? Is that tight? Is it going to be the case that I can explain G with exactly its rank number of rows? Well, yeah, actually it is. We generated G by P times H. Suppose that H is full rank and rank K, and P is full rank and rank K. So I promise I'm going to just, if linear algebra makes your head spin as much as it sometimes does mine, I will do this quickly. But again, G is the product of two distinct matrices, or of two different matrices. If they're both full rank, if P is full row rank, or full column rank, and H is full row rank, then in fact, that will completely explain, that will give us a solution that uses K haplotypes, and it will match the lower bound. So when is the rank of P times H equal to the number of distinct rows? Again, it's the right, this will all line up perfectly. G will have the right number of distinct, the right rank, which will correspond to the number of distinct rows of H, exactly when P and H are full rank. And I'm not going to go into this in incredible detail, but it turns out that P, our pairing matrix, our explanation for how we pair together haplotypes from H to make G, well, P is actually, it's the node edge incidence matrix of a random graph. Why? Because every time I pick two things and put them together, I pick them uniformly at random. That's just picking two nodes and drawing an edge between them. So suddenly a mating is just an edge in a random graph. Well, if you look at a random graph's node edge incidence matrix, that's full rank exactly when it's non-bipartite, and when every connected component of it is non-bipartite. And that's true exactly when you have at least little omega of 1/2 k log k distinct edges. So if you have at least that many rows in G, then you've got full rank. And H is full rank. That was the other part of our product. exactly when, well, that's hard to explain, but it certainly is full rank when you've got a tree. So if you started off with that simple model of evolution where things descend from a common ancestor and a particular position in a sequence has a 1 there if it inherited from a mutation and doesn't if it didn't, well, then in a tree it actually turns out that's full rank too. And the simple explanation for why, which I can show you a little bit more easily on paper, is I can give you a simple explanation of how you can keep pulling one unique haplotype out of H, and it will be linearly independent of everything else. And you just keep doing that until everything's gone and it all works. So what that means is that if you have 0.5 k log k more than that many distinct pairings, then in fact the rank of G is the optimal answer. And so what that means is that this problem is NP hard, but in fact you can solve it with a polynomial time algorithm, at least to compute the number of haplotypes you're looking for, which is really kind of surprising. And you can be a little bit more painful and actually even give an algorithm for doing this when the number goes from 0.5 k log k to maybe 6 k log k. But still, not so very many pairings in your population G mean that you can actually solve this problem in polynomial time. This doesn't explain why the integer program solves quickly. This is an NP-hard problem. We're showing a polynomial algorithm for many instances of the problem. Everywhere it says almost surely here in red, that's the technical term for almost surely, that says with probability very, very close to 1. It doesn't actually explain why the original algorithm works, but it at least gives an explanation that works for why it could be possible to do this. And kind of striking that. So you can actually, that's the end of the algebraic pain. And then just to step away from that, you can also actually solve a lot of other problems in the same domain. Many of these problems are NP-hard. All of them matter, again, because they all give a different way of abstracting away this question of run over? I've gone poof. Of explaining away of explaining your problem in a simple number of steps, and all of them are NP-hard. All of them turn out to be easy to solve in practice, which again is really kind of surprising. And for problems other than the simple one I just told you about, we're working on why it is that they solve rapidly as well. Where do we go next? Well, the general problem that I'm working on here in this whole domain is why is combinatorial haplotyping an easy problem to solve? All of these problems, you can state them, you can solve them, and they're all NP-hard. And they all the reason why they're easy to solve must go back to something having to do with population structures. And so the question is, can we actually give, and typically when you solve them, they not only are easy to solve, but they actually tend to give the answer you started off with. That is, they solve the right answer, which is a bit of a surprise too. So this suggests that maybe one could also approximate the statistical objectives that I said were painful and hellish, and maybe get a good solution that way as well. In fact, you can even go one step further. When people have studied association mapping, this question of relating diseases to genetic variations, that's always been done statistically as well. And an interesting question is, can you study that from a combinatorial point of view too? Again, you might get simpler problems that you might be able to then actually solve as compared to the fuzzy, awful, unpleasant statistical problems that you might want to solve but would never actually be able to finish. So that's the end of this talk. I think the biggest take-home message that I want you to leave with is that understanding genetic variations among people is actually important. It's important at the level of understanding how diseases are identified with their genetic bases. It's even important at a very microscopic level of trying to prevent things like very easy ways of preventing people from dying. You need to be able to, in order to do that, you need to be able to separate paternal and maternal chromosomes. And doing that in a combinatorial domain can be very easy and even give some pretty delicious theoretical results as well that may be extensible to other domains. I just want to finish up by thanking Dan Gusfield, who's actually my host at UC Davis, for introducing me to this whole domain. Many of you may have read his quite well-known book on sequence analysis, and he's just a terrific guy. And the work that I told you about the structure of the haplotyping problem is joint work with one of my graduate students. And my being able to be here is thanks to NSERC, which is the Science Research Council for Canada. Thanks very much. Yeah.
So what's the algorithm?
What's that?
What's the algorithm?
So the algorithm for solving the problem when you don't actually, when you have even more pairings than you start off with. So actually, I mean, it's really a cheat. What you get to do is you get to show that a particular algorithm that was presented in 2004 that has exponential runtime, the runtime of that algorithm is polynomial times the number of different explanations that can exist of a particular type. And what we can show is that the number of explanations is 1. And so consequently, you wind up with a very simple algorithm that's already, in fact, even been implemented, and you just say, I know this is going to work in very fast time. So it's even a cheat. Although you can also turn that around and say that we've, in fact, shown why another algorithm even runs very quickly. But no, ironically, the work that we actually did on this problem, we have another paper that actually works on the integer programming for this problem so that it's not exponential in size. But most of the work that we've done on this problem is actually about explaining why other algorithms are the right things to be using, just from this mathematical structure point of view, which still feels good. Yeah.
You had in the talk yet almost surely and things like that. So obviously there's a probability mechanism you're talking about.
That's right.
Based on which that's happening. And then you talk about NP hardness, but that's really a worst case analysis, not a probabilistic thing. So how do you...
Right, so the question is how to connect the fact that NP hardness is a statement about worst case results and probabilistic analysis is a very different domain. That's true. And I mean, it's not like this is a domain that's particularly, or a phenomenon that's surprising. Lots of NP-hard problems solve very quickly. Satisfiability solves very quickly, traveling salesman problem solves very quickly, knapsack solves very quickly. A lot of these problems have that kind of structure. And indeed, it is the case that, I guess the way to saying that it's a little surprising in this domain is that typically when a problem is easy to solve in practice, but NP hard, usually you have some kind of approximation guarantee algorithms or something else floating around in the literature as well. The approximation guarantees for this problem are appalling. They're all exponential. They basically, in fact, start off by taking the exponential size integer program and then doing randomized rounding on its result, which could raise you up by an exponential factor. So they're just awful. So the thing that's a little weird about this problem is that for one thing, that the very first trick that somebody tried for this problem actually worked, which is a little weird. And also that, unlike a lot of other problems that are NP-hard but easy to solve, there's no approximation results. But yeah, you're absolutely right that I mean, if you look back to the 80s, one of the things that was true in the 80s was that there were a lot of papers showing that NP-hard problems had very easy algorithms when they came from very easy distributions. Here, another thing that's different is that this is an NP-hard problem that has very easy instances when you take them from good distributions, from distributions that are what biologists say is their best model of evolution. And in that domain, we can still say, yep, if you run this, it's going to run in polynomial time. And that's a little surprising too. But you're right. I mean, there's obviously a tension between those two different, quite divergent disciplines for algorithm analysis. Yeah.
Has it been proven that haplotype is more than genotype in terms of prediction?
Yeah, the question is why should you care about haplotypes? Aren't genotypes good enough, in some sense? And I mean, so people are still arguing about this. The HapMap project, which is the project that actually first identified, that was completed last year, that identified, well, actually, it identified genotypes for about 270 people around the world, and then used different software kind of to solve a problem similar to the one that I presented today to infer haplotypes. So that's the best state-of-the-art stuff that we have in this discipline right now. It's not the case that there are certainly a fair number of domains in which, or of diseases in which it's clear that knowing what came together on a chromosome actually gives you a lot more important value than just having one or the other. Rather than just having the ambiguity found in genotypes. It's not the case yet that it's a total win, but it's clear that that's going to be very important. In particular, because certain genes will be tracking also with the promoter regions that govern their expression. It's very clear that getting that association correct, getting that inference correct is going to matter a lot. But you're right, it's still, I mean, we say this as though, of course, it's obvious you're going to want to do this, but with the exception of things like, well, the transplantation rejection case, it's not as cut and dried yet. Yeah? Right, so if you know family structure, this problem is vastly easier. If you know family, so the problem that I presented to you was, here's these genotypes. These people are, I don't think they're related. If you know that this person and this person are the children of this person and this person, even if you don't know the parental genotypes, the problem is vastly simpler. And in fact, that allows you to do much better and much faster analysis to solve it.
So in reality, is it the case that people who need transplants don't have this information, or?
No, and in fact, of course, that's the reason why one solves the haplotyping problem for people who are transplant patients differently. Although it can be the case that, so it is actually, my understanding is that, I mean, this domain, of course, changed dramatically in the last 5 years once easy DNA sequencing became possible, but it is apparently very helpful if you can actually have parental and paternal and maternal DNA, which isn't necessarily possible for a transplant patient who might not have siblings or parents. So you're right that, and so one way of also addressing this is you can haplotype gametes, which is to say you can take a sperm or egg cell sample of a person, and then that's a haplotype, and that, because of recombination, the problem isn't trivial, but you can still use that as a way of mapping back to try to identify what their haplotype is.